We are going to have now the talk by Talía Berruga Fernández, which is going to talk to us about chromosomal hybrids, what these are, and how do they form, and I guess also how do they might be related to antibiotic resistance somehow. Talía, thank you so much, and the stage is yours.
To turn it on. Okay, hi, can you hear me now? Yes. Okay, hi, Eva, thank you. So, thank you everyone for being here today. I am a PhD student with Darmy Hughes lab, and we study chromosomal hybrids. So what I'm gonna be talking about today is what they are and how they are formed. So a chromosomal hybrid is a strain that in its chromosome has foreign DNA. So this means it has a part of the chromosome that is coming from a different strain. This extra DNA has to be integrated into the chromosome. Otherwise, it is not a chromosomal hybrid. And we became interested in hybrids because these two pathogens were found. So one is a Klebsiella pneumoniae, and this strain has or underwent a recombination event between the ST11 and the 4442, and it created a hybrid. And then this hybrid acquired more foreign chromosome from ST42. Here they also show that it acquired another mobile genetic element, and then the other pathogen is an E. coli, and this pathogen is a bit special in the sense that instead of having the foreign DNA integrated as one single fragment, like you see here. So here, the red DNA is all in one single fragment. In the case of this E. coli, it contained a lot of small fragments distributed along its DNA. And within those, it acquired mutations in JARE and PARC, which made it very highly resistant to fluoroquinolones. Both of these strains were found to be hybrid a bit by accident. This was not what they were looking for, but then it turns out that they are hybrids. So what's relevant of both of them is that they are very virulent, multi-drug resistant. Both of them are spread worldwide. More than 1 megabase of their chromosome comes from another strain. This is a lot of DNA, around 25% of the whole chromosome. And it is not clear how they were formed in nature. At this point, it was not clear whether this was just a very rare event or if we could find more of these strains in nature. So for a hybrid to be made, there needs to be transfer of DNA between strains. And this is something that we know that bacteria do relatively frequently, or very frequently actually. There are several mechanisms for this. This is known as horizontal genetic transfer, and it can be either carried out by viruses. These are phage. This is called transduction. Or sometimes when a bacterial cell dies, it releases its DNA out into the environment, and then another cell might take up this DNA, and then we also have conjugation. Now, we focus on the study specifically of conjugation, hybrid formation through conjugation. And for this to happen, the bacteria has to not transfer the plasmid that we see here, but transfer its chromosome. Because we're talking about chromosomal hybrids, so what needs to be moved is the chromosome, not something like a plasmid. So how do we go from a bacteria being able to transfer plasmid to a bacteria being able to transfer its chromosome? We have something that is called formation of HFR strains. So, for example, if we have a bacteria that contains a plasmid, this is shown in black here, and then this plasmid shares some sort of sequence that is similar to what you can find in the chromosome, this plasmid can integrate into the chromosome, for example, through homologous recombination, and then it would look like this. So now the plasmid here is part of the chromosome, but the plasmid still has the ability to carry out conjugation. So when it decides that it will start conjugation, since it is now linked to the chromosome, it will drag with it the chromosomal genes. So this type of strain is called an HFR strain, a high frequency of recombination strain. How much of this chromosomal DNA is going to be transferred into a recipient mostly depends on how long this connection between cells lasts. So the longer it lasts, the more DNA it can transfer. And in theory, if this lasts long enough, you could transfer the whole chromosome. So this mechanism, conjugation, is the only mechanism of horizontal genetic transfer that could allow for something like the hybrids that we talked about before to be formed. I mentioned that they have more than one megabase coming from a foreign strain. Conjugation is probably the only mechanism through which they could have obtained this. Once the DNA enters the recipient cell, which is here shown in blue, then if they have similar sequences, the foreign DNA and the recipient DNA, then again through homologous recombination, this could integrate into the chromosome. For those that do not work with bacteria, homologous recombination is a mechanism that bacteria have to repair DNA. So if anything goes wrong during the replication of the chromosome, they can repair DNA. And what's happening here is that if these two pieces have some similarities, it will repair its chromosome instead of with its own sequence and with the DNA that came in. So then you end up with a hybrid. So then this mechanism of HFR formation, it is known. We know that this happens. And we also know that many clinical strains contain several plasmids. So then we asked, is it possible that clinical strains are usually becoming HFR and then mobilizing the chromosome and creating chromosomal hybrids? And if yes, we wanted to know how often this happens. What proportion of clinical strains out there can do it, at what frequency, how do their hybrids look like, and so on. So to answer all of these questions, we started by doing a screen of a collection of clinical strains. All of these clinical strains were E. coli coming from UTI infections. And we were going to test all of this as possible donors of chromosomal DNA. And then as recipients, we used a lab strain, also an E. coli. But this is a strain that we engineered in the lab, and we inserted this gene here. This will give it resistance to the antibiotic apomycin. And then we deleted one of these 4 genes that are shown in red. All of these genes are part of different pathways to synthesize amino acids. So we deleted one of them in each recipient. In total, we have 4 recipients. And what this happens is that the bacteria can no longer synthesize one of these amino acids. So for example, here, the one I'm showing is if we delete histidine, then the bacteria cannot synthesize histidine, and therefore it will not be able to grow on a culture medium unless it contains histidine. And then what we did was mix these recipients with each of these clinical strains, and then we played on special selective media to select for hybrids. And the way that works is the following. So we have a recipient. I said that we deleted one gene and we inserted a resistant cassette here. And then we mix, so this one cannot grow on a media that does not contain histidine in this case. This is M9 is minimal media. So it's a media that does not contain any amino acids at all. So it won't be able to grow in this media. It will be able to grow in the presence of apomycin. And then if we add the donor to the mixture, this donor, it is not modified by us. So it can synthesize all the amino acids. It will be able to grow on minimal media, but it will be killed by the antibiotic apomycin. And if the donor, which is our clinical isolate, transfers DNA into the recipient. And then this DNA happens to be this little part that we deleted. It can be integrated into the recipient's chromosome. Now we have a hybrid because we have chromosomal DNA that's coming from two different strains. So now we have a hybrid, and this hybrid can grow on minimal media and on apomycin. So if we mix them together into one plate, this is the media that we can use to select for hybrids. So we mix our strains, we let them grow overnight, and then we plate on this special media, and we will only see hybrids growing here. If we find any colonies on these plates, we can take them and we know that they are hybrids. And then we want to look at these hybrids and see which genes they acquire. So these were the results of our screen. We tested in total 118 strains, and 40% of those produced hybrids. So here on the y-axis, I am showing frequency of hybrid formation. This is calculated by dividing the number of hybrids that we obtained on the selection plate by the number of recipients that we used. And then, for example, here, what this number means is if I look into 1 billion cells, I'm going to find one hybrid. So 1 times 10 to the minus 9. And we see that the frequency of, so each dot represents a different donor tested, and the frequency at which they produce hybrids varies quite a lot. Down here, we see which marker we were selecting, TRPE and HEC. We tested the whole clinical the whole clinical isolates collection with these two. And then a subset of those we also tested with this. So then we cover the whole chromosome. We see if they can transfer any marker along the whole chromosome. Yeah, and then we just see that we have different frequencies for each one. Just as a comparison, the frequency of spontaneous mutations at the single nucleotide level is around 10 to the minus 9, 10 to the minus 10. So around this area here, we have things that are more frequent, things that are less frequent. Then we want to analyze those hybrids that we obtained. To do this, we do whole genome sequencing of the hybrid, and then we need to compare this hybrid to both of the parental strains to know what part comes from where. So I align it to my recipient DNA. This is MG1655, a laboratory strain. And I say, oh, okay, everything here is identical to my recipient. But then I have this weird gap that looks completely different. So if I want to know whether this came from my clinical strain, I need to also sequence my donor, come up with a reference genome for my donor, and then I use this reference to compare it to the hybrid. And hopefully I can see that this gap that I had before is identical to the donor. And this way I confirm that what I isolated is indeed a hybrid, and I see how it looks. So by this analysis, we can come up with maps that look like this. What we see here at the top is a genetic map of our recipient. This is where we have our resistance cassette. And then all of these are the genes that we deleted in the recipients, which is what we're selecting for. We're selecting for transfer of one of these. And here in different colors, I show which of the markers is being selected for each strain. So we just look at the size of the fragment, where the fragment sits in the chromosome, how many fragments we got. Sometimes we get one, sometimes we get more. Total amount of DNA transferred. Did anything else come in with this DNA? So here, for example, we have plasmid number 4 from this donor also coming into the hybrid. So there's a lot that I could say about this, but the one thing that I'm going to focus on right now is that this donor, the one that I'm showing here, has 7 plasmids. And we want to know which one of these plasmids is the responsible one of mobilizing the chromosome. Is it any of them, all of them, only one of them? We have 3 of them which are unmobilizable, so it's very unlikely that these are the ones, but we have 4 that are conjugative. So any of these 4 could be the one mobilizing the chromosome and creating these hybrids. So then what we did to try and figure out what was happening is the following. We start again with our clinical strain that has several plasmids. We do conjugation experiments again, but this time selecting for something that is in the plasmid. So in this case, I'm showing that this plasmid had a gene that made it that confers resistant to trimethoprim. So if I mix it with a lab strain that is resistant to apromycin, and then I select in media that contains both trimethoprim and apromycin, I can select for the transfer of this plasmid. And once I have the plasmid in my strain, I get rid of this apromycin resistance. And now this strain, I'm going to use it as a new donor to test whether this plasmid can on its own mobilize the chromosome. So then I use this as a donor, again in conjugation experiments with the recipients that I have shown before, the ones where we delete one gene to make them not be able to grow without this antibiotic. This is called oxytrophic. And hopefully we get hybrids. I also do this experiment in using MG1655 as a donor. So this strain, but without the plasmid. And then I show that I don't get any hybrids. But if I have the plasmid, then I get hybrids. So our ideal new donor would be an MG1655 background. So the chromosome is MG1555, and it only has one mobile genetic element from the original clinical strain. So in this case, we have only one plasmid. Through this method, we isolated as many plasmids as we could from the clinical collection, and we got in the end 10 plasmids from 7 donors. We tested all of them, and it turns out that all of them were able to mobilize the chromosome. As long as they're conjugative, they can mobilize the chromosome. And here I'm showing the frequencies. I am not showing the frequencies for the 10 plasmids, so instead what I'm focusing on is 2 different plasmids coming from the same strain. So one clinical strain is represented in blue. We have tested 2 plasmids from that same strain. Another one is green and another one is yellow. So whatever plasmids we select, if they are conjugative, we can mobilize the chromosome. For these 2, I didn't get any hybrids with this marker, but with other markers, we do get hybrids. And obviously, this one plasmid stands out because it has a much higher frequency, almost 10 to the minus 6 or above it. And that's quite high. I want to also point out that the frequencies here, if you notice, are in general higher than the ones we obtained from the original screen that we did. And that is most likely because now we're using 2 strains, both donor and the recipient have identical sequences in the chromosome. So the closer they are, the more likely they are to form hybrids as well. And then coming back to these maps, these are the hybrids from a different donor. And what we noticed in this case was that almost all of the hybrids contain this yellow thing that I have marked here, which was a genomic island that was always present in one of 2 genes. They are in tRNA genes. It was always either in one or in the other or in both. And we wondered, what is this thing that is always being transferred? And it turns out that it is an integrative conjugative element, or an ICE. What ICE are is a little fragment of DNA that sits in the chromosome, and it has the genes to initiate conjugation. It has all the genes needed for the conjugation machinery, the same that the plasmids use. So it can transfer between cells. It usually comes out of the chromosome. It forms a circle. It makes a copy of itself, and then it transfers to another cell. So this is kind of like exactly what a plasmid would do. And once it's in the new cell, it can integrate in the chromosome of that recipient. and the copy that was left back goes back into the chromosome of the original strain. So because it was transferred all the time, and we know that it has the genes to carry out conjugation, we wanted to know, is it possible that this is also mobilizing the chromosome? So we did the same that I had described before, but this time we're selecting for the transfer of the eyes. So I do conjugations. The eyes have trimethoprim resistance in it. So I could select with the same antibiotics, trimethoprim and apromycin. Once I get the eyes into the lab strain, the MG1655, I get rid of the apromycin resistance. We sequence this strain to make sure that no plasmids came with it. We only want to test whether the eyes can do it. And then we do conjugations again with the recipients that we always use. And what happened was that, yes, indeed, the eyes can mobilize the chromosome. So here, the strains that contain an eyes are marked in either a triangle or a square, depending on where the eyes is sitting in the chromosome. You can see here where they are, where each of them is. And all of them could, sorry, it's two eyes. Both of them could transfer the chromosome. And in general, they do it at a way higher frequency than the plasmids that we tested here. And then since we are part of the UAC, I'm going to come back to how is this all relevant to antibiotic resistance. So here we have again these maps, and when we look at all the things that were transferred from the donor into our recipients, part of the analysis is to look at what genes are included in all of this. And it turns out that very frequently it is genes that are associated to antimicrobial resistance. And this can be of different types. It can be, for example, mutations in genes that sit in the chromosome that confer resistance, like mutations in JAR-A. So the recipient, the original recipient, is sensitive to fluoroquinolones. But if it acquires a mutation in this gene from the donor, it is now resistant to fluoroquinolones. We also have completely new genes that did not exist in the recipient that can modify antibiotics and this way inactivate them, such as the beta-lactamases. And we also have mutations in different efflux pumps that could contribute to an increase in resistance. And of course, together with all of this, we very frequently find that virulence genes were also transferred. So here, I mean, this is, I think, 3 megabases of chromosomes. So it's a huge amount of DNA that is being transferred between cells. So you can get a lot of things into your recipient. And of course, here we're going from a clinical strain into a lab strain that has no resistance whatsoever, no virulence whatsoever. But if this happens between 2 clinical strains, then they can sort of combine their virulence profile and resistance profile and come up with something new, which is it's probably what happened with the pathogens that I talked about in the beginning. So just as a summary of the whole project, we have seen that approximately 40% of the strains that we tested could mobilize the chromosome, and the frequencies can be as high as 10 to the minus 6 if the strains are closely related. All conjugative plasmids that we tested could do it, and we showed that other mobile genetic elements can also do it. This is the eyes that we showed. And resistance and virulence determinants are frequently transferred into these hybrids. So thank you for listening, and thank you to Darmit's group and everyone else that has contributed in this project.
Thank you so much, Talia. Thank you. That was great presentation. Very good to see how the project is going. I think Gerhard might have a question for you because he opened up his camera. So Gerhard, go ahead.
Okay. Yeah, I have one question. Can you hear me?
You, I think. Or yes, we can.
Yeah.
Sorry.
Okay. So my question is the following. I mean, in the cases where you find these multiple inserts, I mean, 7 or whatever of the chromosome. Do you believe this is, let's say, one transfer event and then different parts being sort of recombined in separately, or is that more likely to be several transfers that result in this patchy pattern?
So based on frequencies, I think it is more likely that it was one conjugation event that led to many patchy, so many homologous recombination events at the same time. Because for them to be conjugating many times, I mean, the plasmid first has to integrate into the chromosome, right? And then transfer the DNA. So then the frequency of that is already relatively low. And for that to happen in the exact same cell many times, I think it's more likely that once it happens and the DNA in the recipient starts to be degraded, then you get many recombination events.
Okay, I understand, but in that case, I guess the follow-up question would be, why do you see that in certain combinations of strains or wherever? In a few strains, you see these very patchy patterns, but most of them seem to have one chunk. Is there anything different in the machinery that's present in the recipient or so, or what is the most likely idea?
Yeah, so we noticed the same, that depending on which donor you use, you get this sort of different pattern. Some donors very consistently give you a very small fragment of DNA, and others give you a lot and very patchy.
Okay.
It's not clear for us what it is doing it. We did some experiments where we modified the recipient. We deleted the restriction system. We deleted what else did we delete? Mismatch repair system.
Yeah.
To see if we get any difference in the pattern, and we always get the same depending on donor. So it seems like it's something coming from the donor, not something present in the recipient, at least. But what it is, it's not clear. No.
Okay. Thanks. Great story.
Yeah, it's really cool. Actually, following up on Gerhard's talk and what you're talking about, that it's certain donors tend to have the same patterns, because you also from certain donors took away the elements that were making the hybrids and put them in a lab strain and check it. Did that pattern continue once that mobile element was put on a lab strain background and then used to make the hybrids or not?
So the thing is, let me see if I can show.
When I did those experiments, both my donor and my recipient are identical. So both chromosomes are identical. So I cannot do whole genome sequencing and see which parts of the chromosome came. I just know that this was fixed because it now grows in my selected media. What I have done is, instead of using MG6055 as a
recipient here, is used a different strain, an ATCC, and then the chromosomes are different enough that I can actually sequence and look at them. In that case, I only get hybrids when I use the eyes, and the pattern between that and the original clinical strain is similar, a little bit different, but more or less similar.
All right, and now I understand. Great. Let's see if anybody has some questions. I have tons of questions, but let's see, maybe people will think some more questions and we can have a little bit of a discussion regarding I like that you're looking into, what this could be the implications when it happens in between strains out in the wild, right? And we're thinking about, in particular, maybe strains that can meet in our guts, for example. So I have several questions that go on that, I think, track. One is, how is this conjugation happening? Does it happen when they meet in a liquid format, in a solid format? How would that actually be relevant to the physiology, how bacteria might find each other in the wild? That's one part. And then the other part is, I did used to study plasmids back in my days of doing research, and I know for a fact that there's some limitations sometimes between which plasmid can be sent to different species even, or between different strains. which is called the host range, right, of a plasmid. So now it came to me the question of what are the host ranges for these ice elements that you are showing they can actually form hybrids? Do you know if they can be sent to same species, between different species? Is there any limitation to this? If you can tell us a little bit about that.
So for your first question, the conjugations that we do are on solid media. The reason behind this is that then we increase the efficiency of conjugation a lot, and we can increase the amount of cells that we look at a lot. So then our detection limit of hybrid formation is very, very high. Like we can find hybrids that are formed at a very low frequency. In nature, if this, for example, happened inside a patient, then it would likely be in the gastrointestinal tract. So that is more of a liquid media, right? But then the numbers that you're dealing with in a person would be most likely much higher. And then for your second question, eyes, as far as I know, have quite a broad range. We haven't tested this in particular that comes from an E. coli, but there are papers transferring eyes from other species into E. coli, and we know, for example, that Klebsiella can transfer plasmids into E. coli, so it is very likely that eyes can also mobilize between different species.
Very, very good, very interesting. So, what is you guys' hypothesis on why has this not been studied before? Because I know this is quite recent. I mean, we know about hybrids, but look at it systematically, especially within clinical strains where the transfer of genetic material is of special importance to antimicrobial resistance. Why has this kind of been under the shadows for so long?
Well, the hybrids that I talked about in the beginning, the ones that are pathogen, it was kind of an accident that they were found to be hybrids. If you take a strain and sequence it, you see, a string of letters. That doesn't really tell you anything unless you happen to have the reference of the parental strains of this hybrid. And that's what they had. They were lucky to have many DNA sequences, and when they compare them between them, they realized that, a part of this looks like this strain and another part looks like this other strain.
But unless you
Have the sequence of the parentals, you will not realize that what you're looking at is a hybrid.
Yeah, you kind of need to kind of see for that in particular. So Lisa is asking you, what would you do next?
So there are still several questions that we want to answer. One of them is what Gerard mentioned. Why do we sometimes see one single fragment, and why do we sometimes see different patches? And there's also studying the interaction between plasmids. So how would the presence of two plasmids affect chromosome transfer? Does it matter which kind of plasmids they are? Here in the experiments that we're doing, we have one donor that has a lot of plasmids and then one recipient that has no plasmids. And that is not what you would have in reality. In reality, you would most likely have two strains and both of them would contain plasmids. So that's another, like a next step to study. If we have a recipient that has different plasmids, how does this affect chromosomal transfer?
Do you think they might actually come that you find that they send DNA both directions?
Yes, we have actually already seen evidence that that can happen, that it can go in both directions. We also have experiments planned to study this. If one strain has a plasmid and this plasmid is transferred into the recipient, can then this recipient be the new donor and transfer DNA back? Or can it go simultaneously both ways? Yeah.
This is so fascinating. I love it because also there's so many implications to the way we also see kind of bacterial evolution as a whole, because even if we actually thought already that, evolution doesn't really look fully like a tree with branches for bacteria, because you don't only have vertical inheritance, but also horizontal through plasmids. If now it turns out that it's not only plasmids, but it's also kind of the chromosomes with plasmids in it, that they can go both ways. It's a super fluid this landscape of genes moving from one way to another and then being shared between strains. So it opens up like a huge outlook into how bacteria might evolve, and obviously also for selection.
Yes, definitely. I mean, if both of the strains that you have together in a population can potentially transfer their chromosome, then the combinations that you can get are huge. And in the hybrids that I showed that acquired resistance determinants, we did not select for any of that, and it's gonna be stable because it sits in the chromosome and it is E. coli DNA in the end. So they're not gonna try to get rid of it. They can grow happily with it, and if there happens to be a selection pressure at some point, they are gonna grow better than the parental.
Yeah, I was going to ask some questions about that because, yeah, you're looking into what kind of resistance might be in the transfer part, but that's kind of like a byproduct of just the transfer happening because they are strains that might have more resistant determinants because they have been evolving in a clinical setup. But it's not really that they are going for it or need it for in any way until in the future they might encounter an environment with antibiotics. So it's really cool. Anybody has any further questions? I love evolution so much. I think this is a great story, very cool, and very. What I love also about these kind of projects is that the essence is very basic research, but the possible implications on topics are there, and could be developed further to look into particular setups. So it's really really cool. If nobody has any further questions, then I think we might be closing up the first webinar of this 2025. I will thank you both, Talía and Shiwo, for presenting your research. It's really cool to see how the projects are following up and developing and forming to be, published research and on the way to your defenses. And all of you out there that have been attentive to these two talks and stay with us for these 1.5 hours. And I hope to see all of you at the next webinar, which will be on the first Friday of March. We will have another 2 PhD students. And also just to remind you that our website calendar is up to date. So you can go in and you can check what events have been already set up for UAC, and you'll find more information if some events might need registration or some details of what is going on with that. Apart from that, I just wish you a really great weekend ahead and to have you at the next event.